Welcome back to the All Sorts podcast for another deep dive. We love digging into fascinating research, and today, well, we're heading into the deep ocean.
We are indeed. We're talking about something truly mesmerizing. Bioluminescence. Living light in the sea.
Exactly. The science and the sheer wonder of it. We've got some great scientific papers guiding us, looking at how it works, why it happens, and where you find these glowing creatures.
And it's surprisingly common down there, isn't it?
Yeah.
Especially in the deep sea.
Way more common than on land, yeah. The sources suggest maybe over 90% of deep sea life might glow.
Incredible. Just think about that. A world lit from within, completely different from up here.
So why the ocean? The research points to maybe a couple of things, like just the sheer amount of time life's had to evolve underwater without interruption.
And the environment itself, right? Huge areas of total darkness.
Yeah.
Perfect stage for light signals to become really important.
Totally. So let's dive in and illuminate this glowing story from the basic chemistry up to the big picture.
Sounds good. So at its core, bioluminescence is actually, well, it's a chemical reaction. Visible light produced through oxidation.
Okay, oxidation. And how do the animals actually make the light?
It varies. Some have pretty simple light-making cells called photocytes.
Photocytes, got it.
Others have evolved these really quite complex structures, photophores. Think tiny biological lamps, sometimes with like lenses or reflectors built in.
Yeah, like mini lighthouses on a fish. That's amazing. The papers also mention symbiotic ones.
Ah, yes. That's a whole other level of cool. The animal itself doesn't make the light. Instead, it hosts colonies of bioluminescent bacteria in special organs.
So the bacteria glow, and the animal uses their light, like little lodgers paying rent with light.
Pretty much. The bacteria has a safe place to live, nutrients, and the host gets a controllable light source. A neat partnership.
Incredible. So how do they control it? Is it just always on, or?
Oh, no. Definitely controllable. It's often switched on and off using physiological triggers, like neurotransmitters, hormones.
Like a biological dimmer switch.
Exactly. Allowing for everything from really short flashes, less than a couple of seconds,
Right.
to much longer, sustained glows. It's quite sophisticated control over that chemical reaction.
It really is. One thing that jumped out was about dinoflagellates.
Yes. The single-celled algae.
Yeah. And apparently you can actually buy live ones. Pyrocystis fusiformis, people use them in aquariums, art educators.
You can. It's
Yeah.
amazing. Brings that little bit of ocean magic right into your hands. Imagine seeing that glow when you disturb the water.
Like holding stars. It's
Yeah.
incredible. Okay, so that brings us nicely to the microscopic light sources. Let's talk about the bacteria themselves. The sources call them mesmerizing luminous organisms.
They really are. They have their own fascinating story.
So how do these tiny things manage it?
Well, it's down to specific genes, the Lux genes. They control the light production.
Okay, Lux genes. Makes sense.
But the really clever part is how they control it. It involves something called quorum sensing.
Quorum sensing. Sounds like they're taking a vote.
Sort of. It's about density. A few bacteria on their own won't glow much, but when enough of them gather together, they release signaling molecules. Once a concentration of those signals hits a certain point...
A light switch on?
Exactly. They collectively activate the Lux genes and start producing that typical blue-green light, usually around 450 to 490 nanometers.
That is clever. A group decision to glow. And this isn't just for show, right? I saw mentions of practical uses, pollution monitoring.
Precisely. That's a big area of research. Scientists are looking for naturally super bright strains to use in bioassays.
Bioassays, like using the bacteria to test water quality.
Yeah. If pollutants are present, they can affect the bacteria's light output, signaling a problem. There's a strain mentioned, Vibrio campbellii LZ5, showing promise for detecting heavy metals.
That's brilliant. So you can use the whole bacteria.
Right, as living sensors. Or you can actually extract the enzyme responsible for the light, the luciferase, and use that as a biosensor in lab tests for toxins. Really useful in biomedical research and toxicology, too.
Amazing versatility from these tiny organisms. And one study really went deep on a specific strain, didn't it? Vibrio campbellii, STF1. Found on a stonefish.
Yes, they did a very thorough workup on that one. Noted its really intense blue glow, especially when they grew it in a liquid broth.
So what did they find out about it?
Well, they characterized everything. It shaped short little rods that can move, ran all sorts of biochemical tests, looked at its unique fatty acid signature, even used a technique called MALDI-TOFIF.
Okay, fancy stuff, like getting its protein fingerprint.
Essentially, yes, identifying specific protein markers unique to it. They really wanted to understand this bug inside and out.
And what about its preferences? Like, when does it grow best and glow brightest?
They figured that out too. It grows fastest in the first 6 hours or so, hits peak luminescence around 8 hours, and seems to like a slightly alkaline condition, pH 9.
Interesting. And did they test how it reacts to things like antibiotics or metals?
They did, rigorously. Tested against a panel of antibiotics and different heavy metals.
Any surprises?
It was sensitive to most antibiotics, but interestingly, resistant to a couple, like amoxiclave and ampicillin. For metals, it didn't like mercury or cadmium, but it wasn't bothered by lead.
Huh. Resistant to lead. Wonder why. And they even identified the light-making enzyme parts.
Yep, the luciferase subunits. Found they had molecular masses of about 40 and 37 kilodaltons, really getting down to the nuts and bolts of the light production.
It sounds like it, but there was more, wasn't there? Something about antibacterial activity.
Yes, another really interesting finding. Extracts made from this STF1 strain could actually inhibit the growth of several human pathogens.
Seriously? Like what?
The strongest effect was against Shigella dysenteriae type 5, which causes dysentery. They even used GCMS analysis.
Gas chromatography mass spectrometry to find out what chemical is doing it.
Exactly. They pinpointed a couple of compounds, tetradecanol and something called 2-tert-butyl-4-isopropyl 5-methylphenol, as the likely candidates for this antimicrobial action.
Wow, so it glows and it makes potential antibiotics. This little bacterium is full of surprises. Did they look at its genes too?
They did. Genetic characterization confirmed it was Vibrio campbellii, finding the expected Lux-A gene for luminescence and another gene called Toxar. The sequences were very similar to other known strains.
Okay.
They also found plasmid DNA.
Plasmids, those extra bits of circular DNA.
Right, and they think that might be where its antibiotic resistance comes from, potentially.
So pulling all that detail on STF1 together, what's the big takeaway?
Well, it really highlights how these natural bacterial strains can be incredibly effective biosensors, often glowing brighter than engineered ones. Plus, discovering those phenol compounds opens doors for potential new drug development.
Right.
And it shows there's still so much to learn about the genetic diversity of these bacteria in places like the Andaman Islands where this one was found. Needs more research, like whole genome sequencing.
It's humbling, isn't it? All this complexity packed into something microscopic. Okay, let's zoom out a bit. From the tiny bacteria to the bigger picture across the whole ocean, another source tried to catalog all the known glowing marine animals. Sounds like a massive job.
Oh, absolutely monumental. They had to sift through, like, over 1,000 publications, plus unpublished notes, and deal with the fact that scientific names change over time. Systematics is complicated.
I can only imagine, like trying to assemble a constantly shifting puzzle. How many did they end up cataloging?
They documented reports for 819 genera and nearly 3,000 species, 2,951 to be exact.
Wow.
And importantly, they graded the evidence for each one, used a 1 to 6 scale for how reliable the report of luminescence was.
So a confidence score for the glow.
Pretty much. And the good news is most of them scored highly. Over 87% of the genera and 95% of the species got a score of 4 or more, which they classified as definitely luminescent for their analysis.
Okay, so a solid list of confirmed glowers. What about the maybe glowers, the ones needing more proof?
Yeah, those are interesting too. They flagged about 99 genera where the evidence is weaker and needs follow-up. But if you combine the confirmed and the potential ones, it covers over 9,400 species.
1,400, that's huge.
It is. But here's the flip side. They only found about 99 species within those groups that are definitely not luminescent. And for almost 68% of the species in those potentially glowing groups, the status is just unknown.
68% unknown. So vast swaths of the ocean's biodiversity might be glowing, and we just haven't seen it or documented it yet.
Precisely. It points to a huge potential for discovery, especially in the really deep parts of the ocean that are hard to study.
Makes sense. So how is this glow distributed? Are some groups just packed with luminescent species?
Definitely. Looking at the number of genera with glowing members, the big three are chordata, that's fishes and relatives, arthropoda like shrimp and copepods, and cnidaria jellyfish, corals, that kind of thing.
Yeah.
But if you look at the sheer number of glowing species, chordata, arthropoda, and mollusca squids, snails, etc., make up over 84% of the known total.
So fishes, crustaceans, and mollusks are the main players in terms of species count.
Right, and some specific groups really stand out, like a very high percentage of species within the decapodiforms, that's squids and cuttlefish, are luminous. Same for ctenophores, the comb jellies.
Ah, so while fish contribute a lot of species overall, groups like squid are almost all glowers.
Interesting. What about other invertebrates?
For most other invertebrate groups, the glowing species are actually a pretty small fraction, usually less than 5% of all the known species in that group.
So it's widespread but not everywhere, and definitely concentrated in certain lineages.
Exactly. And again, that huge number of unknowns suggests we could find many more, especially among things living on the deep sea floor benthic invertebrates.
The deep sea floor, another frontier. I also saw a point that it's rare to find a non-glowing species in a genus that's otherwise known for glowing. Yeah, that's significant. It suggests that once a group evolves bioluminescence, it tends to stick around. It's a resilient trait, evolutionarily speaking, not easily lost.
So it must be pretty useful down there.
It seems so. This uneven pattern we see some groups full of light, others not so much, is likely a mix of real biological differences and, frankly, just where we've been able to look so far. Observational bias is always a factor, especially in the deep ocean.
Absolutely. So much still hidden. This whole field feels like it's just waiting for the next technological leap to let us see more. It's incredible to think about this hidden world of light, from bacteria to fish.
It really is. And as that big catalog shows, we're learning more all the time, filling in the gaps. But the deep sea, it definitely holds on to its secrets tightly. Bioluminescence is one of its most beautiful, most intriguing mysteries.
It really makes you wonder, doesn't it, about the silent light shows happening constantly in the abyss, flashes and glows, like a whole language we're only just starting to tune into. What else is going on down there in the dark?
It's a profound question. The sheer scale of the unknown is humbling. Bioluminescence gives us a tiny, shimmering glimpse into an ecosystem that's still largely alien to us.
Well, like those deep-sea lights, we here at the All Sorts podcast hope we've illuminated something new and wonderful for you today. This dive into bioluminescence has certainly sparked my curiosity even more.
Mine too. Always more to learn.
And there's always more to explore with us. If you enjoyed this, why not dive into our archive of past deep dives? Keep that current of curiosity flowing strong by liking, sharing, and subscribing to the All Sorts podcast on YouTube. You can also follow our explorations and join the conversation on X, Instagram, and Threads. Let's keep the flame of learning alive together right here on the All Sorts podcast.